Hello again, student. Next chapter is the lifespan development, and we're going to focus mostly on early development, because I believe that early development determines much more what goes on in the individual for the rest of their life. So this is a summary of what we're going to discuss. We are first going to talk about methodology in the study of development, the nature versus nurture controversy. We're going to discuss prenatal development, the theories of Piaget and Vygotsky, focusing on the development of cognition and the development of attachment. There are two main methods for studying development that are specific for development, and they are aside from the regular methodology that we have discussed earlier in chapter 1, when we discussed, for example, the differences between different methods of observation, differences between a correlational study and an experiment. Whenever researchers are interested in development, they can either use and adopt a longitudinal or a cross-sectional study. And well, basically, first we have to discuss for a moment what is the study of development. Study of development is studying changes that occurs, that are observable or measurable in individuals over time. So what happens to individual over a determined period of time, possibly even the entire lifetime. So we can study changes over a period of time by following the same subjects, and that would be a longitudinal study, or we could employ a cross-sectional study which uses different subjects. Now, the best method, of course, would be a longitudinal study where you're studying the same individuals across a certain amount of time. So let's assume you want to measure and study the different memory abilities in children between, let's say, the ages of 3 and 10. What you could do is that you could measure the memory ability of the same group of subjects when they're 3, and let's assume that you want to test them every year, so you would do it again when they're 4, do it again when they're 5, and so on. Now, that study, just to gather data, would require at least 7 years to be conducted, right? If you're following this individual between the ages of 3 and 10. Now imagine when you're conducting a study that might even focuses on a larger time span. Maybe you want to figure out, changes that occurs in a particular psychological phenomena between the ages of 15 and 30, right? That would be a 15-year study. So longitudinal study takes a long time, and sometimes they're not always possible. Now, the alternative to that is a cross-sectional study where you're using different subjects. So in the first example I gave you before, instead of waiting 8 years and studying these children when they are 3, 4, 5, 6, 7, 8, 9, and 10, what you could do is you can get different subjects, a group of children that are 3, and you measure their memory ability, a group of students that are 4, and you measure memory ability, and so on. So you could do that at the same time. The only problem, though, is that you're studying different subjects, so you can never be sure in a cross-sectional study that the results that you're obtaining are due to changes in development, because the changes could be due to changes in the ability of a group of subjects, right? So it could be due to, let's say you find that there's no difference between the memory abilities of 4 and 5. It could be that differences could be caused by the fact that the group of 4-year-old that you are studying had a much better memory than the normal individuals, the normal 4-year-old, and perhaps the 5-year-old had a lower than usual memory ability. Okay, so only by conducting a longitudinal study you can be sure that what you're observing is due to development. Now, one of the greatest controversies in psychology is the nature versus nurture controversy. We have touched on this already previously in a semester, specifically in chapter 1, I believe, and it's the controversy refers to the fact that for some researcher the psychological phenomena that they're interested in are thought to be caused by genes, while for other individuals, for other researchers, the changes that are observable or measurable are due to environmental influences. So nature refers to changes that are due to inherited traits, abilities, and capacities, and specifically as they relate to development, they are thought to be the effects of maturation. So that these genes that we inherit mature, like it will be a fruit on a tree, right? They change due to the passage of time, so that your ability, let's say, increases in your memory performance are due to the fact that your gene changes and you mature. Now, of course, other individuals believe that these developmental changes are due to environmental influences, the environment, in particular learning, that the environmental circumstances that you're exposed to determines, very importantly, changes in your behavior. So in this particular case, for example, a researcher focusing on the importance of nurture would say that a child increases their memory, increases their ability to remember information because of what they learn. They learn that performing particular mental task enables them to remember the information better, right? So well, as we have discussed already, I believe, in reality, whenever you're observing a psychological phenomena, vast majority of the situation, that phenomena is caused by a combination of these two, combination of nature and nurture, combination of, specifically for development, the maturation of your genes, and learning by being exposed to environmental circumstances. Now let's focus on prenatal development. Probably one of the most important and dramatic periods during the development of an individual, because it's where the vast majority of changes occurs. And if you think about it, we develop as human from a single fertilized cell to a fully mature, fully grown baby. So it's a period of time during which we develop most rapidly and most frequently. So there are three main periods of prenatal development. We can consider the first 2 weeks of prenatal development as the period of a zygote, or period of the egg. Then comes the period of the embryo and the fetal period of the fetus. The first period of the zygote, occurs after fertilization. So when the sperm fertilizes the cell, and that cell begins to replicate, copy itself over and over again, and then at the same time that fertilized cell travels down the fallopian tube and is implanted into the wall of the uterus. And I have a picture here to show you how. You can see here we have an ovary, and once a month, as you know, approximately, a female will release an egg from the ovary, and that egg will be ready for fertilization, to be fertilized. Now a sperm is going to travel from the uterus up the fallopian tube and fertilize the cell, the egg, usually in the upper third of the fallopian tube, right? So approximately right there. And when the cell becomes fertilized, then it begins to replicate. First, of course, as you remember, the DNA from the male, the sperm, and the female egg unite from 23 single chromosomes carried by the sperm and the egg. It becomes a cell composed of 23 pairs of chromosomes, and then there's cell division that begins. So it becomes, from one cell it becomes 2, from 2 it becomes 4, 8, 16, and so on. Now, as you can see, as the cell divides and replicates, it travels down the fallopian tube into the uterus, and then about a week after fertilization, the cell begins to implant itself. Actually, the cluster of cells, I should say, it's called the blastocyst, begins to implant itself into the wall of the uterus, and it fuses to the wall of the uterus in another week, right? So a week to travel approximately down the fallopian tube into the uterus, another week to become completely implanted and attached to the wall of the uterus, right? And that's when the next period of the embryo begins, from week 3 to week 8, and this is the most rapidly changing processes occurs during this period. Cells begins to differentiate, and different body structures are formed. Limbs, internal organs, the 3 layers of the embryo develop. Cells begin to differentiate into bones, into skin, into cells that will become part of a nervous system in a structure called the neural tube. And at the same time, during the period of the embryo, the umbilical cord connects the embryo to the placenta. Up to this point, the cluster of cells were self-sustaining, but at this point, because of these rapid changes and the requirement for more energy, some nutrients are required from the mother. And so the umbilical cord is what will allow this developing organism to get the nutrients that are required for such a rapid growth. And finally, the period of the fetal period, week 9 till birth. Week 9, if you remember, approximately there's the beginning of the differentiation of the ovaries and testes, produced by either the presence or the absence of the Y chromosome. If a Y is present in the organism, that organism will turn into a male. If a Y is absent, it will turn into a female by developing ovaries. Week 12, the circulatory system begins to function. Now, even though, remember, the heart itself begins to function, the primordial heart begins to function just days after conception. And around week 16, movement is typically felt by the mother. As you can see here, the fetus, in this case, the umbilical cord, and that umbilical cord connects to the placenta, and you can see this area that's enlarged. And what this is intended to show you is the fact that the blood vessels in the umbilical cord that receives nutrients from the mother do not really touch the blood of the mother. So there's a filter, if you will, in placenta that protects under most circumstances, protects the developing organism from potential harmful effects coming from the mother. And that is what I would like to focus next, which is the potential negative effects produced by either drugs, disease, or environmental hazard that occur during prenatal development. And these they're all called teratogens. Teratogens is actually a word coming from a Greek word that means monster, and that is because some of these effects can in fact cause some physical malformation. And historically, when babies were born malformed with some physical malformation, they were seen as as monsters. So these teratogens, as I was saying, can be drugs. Some of these drugs can actually be just legal drug, like aspirin or caffeine, or some more controlled, like alcohol and nicotine, and some illegal, like narcotics, and another controlled one would be marijuana. So all of these drugs can have negative effects on the development of or prenatal development of the organism. Then we have diseases like AIDS, genital herpes, rubella, syphilis, that can be passed on by the mother to the developing organism. And finally, we have environmental hazards like heavy metal, like lead, mercury. We have pesticides like PCB, and X-rays can also have negative effects prenatally. And as I showed you before, right, in some cases the placenta works very well as a filter to protect the developing organism from harmful effects of these negative stimuli, but in some other cases it does not. Like, for example, we know that alcohol goes through the placental wall, and in essence, when the mother is drinking, some of that alcohol that enters in a circulation in the blood of the mother also enters in the blood of the developing organism. And of course, as some of you know, I'm sure, drinking alcohol during pregnancy can be very dangerous, and baby can be born with fetal alcohol syndrome, which is a situation in which babies are born with some cognitive as well as some physical deficits. Now, another interesting point to consider is the sensitivity from the developing organism to these teratogens, to these substances that can be potentially harmful. And you see in this chart the different period of prenatal development, a period of zygote over here of the ovum, first 2 weeks, period of the embryo up to week 8, and then a period of the fetus. Right? And you see here what the organism look like. Right over here would be just a cell, and then it turns into this wild-looking creature. See that originally we actually have a tail prenatally, right? Then that tail disappears. Interesting to consider and look at the similarities between human embryos and the embryo of other animals. That looks like very, very similar to each other, almost identical. So that tells you about evolution a little bit. In any event, and you can see how, then during the period of the embryo you have development of these major areas of our body, limbs, the brain, and so on. Now, the most telling portion of this chart is what's down here, which indicates the sensitivity to teratogens. In blue you have a greater sensitivity, and in green you have less sensitivity, although still potential harm. Right? So, for example, the first bar over here indicates the effects on the central nervous system, and it shows that for the central nervous system, a greater sensitivity to teratogens occurs from week 3 to week 6 approximately. Then you have a heart, then you have arms, eyes, legs, teeth, palate. Right? Palate needs to be formed properly. Sometimes the palate doesn't close, and babies are born with an open palate at the top. Proper development of external genitalia and the ear. Now, as you can see, these blue lines, these blue bars representing the greater sensitivity and negative effects, occur for the most part when? During the period of the embryo. Now you have to consider that this period of the embryo is only a few weeks after the pregnancy began, right? After the ovum, the egg, was fertilized. So, unfortunately, at this point, a lot of mothers are not yet aware that they are pregnant, right? Because if you consider that the mother got pregnant only, as I was saying, 5 or 6 weeks prior, she might have missed the period, which is not too unusual for a female, right? To miss a period every once in a while. So what I'm trying to say here is that unless you are planning a pregnancy and you want to get pregnant, it's hard to prevent this, some at least of these negative effects of teratogens. So a female who's not planning a pregnancy might still drink wine or alcohol, might still use coffee and aspirin, might still perhaps every once in a while use some illegal drugs that could significantly negatively affect the embryo. So best thing to do would be, of course, to plan a pregnancy so that the female would stay away from these potential harmful effects to begin with. Let's move on to Piaget theory of cognitive development. And Piaget Piaget is a Swiss cognitive psychologist who believed that children develop mentally, cognitively, by going through a sequence of stages. So it's a stepwise process from one step to the next, and they increase their ability so that their understanding of their surroundings become increasingly more accurate and complex. And according to Piaget, children do that by developing schema, which are mental structures used to organize knowledge. And then these schema are used in the assimilation, which is the incorporation of new stimuli into their existing schema, to their existing habit or mental representation. And also schemas are modified through accommodation. So accommodation are new ways of responding to objects in order to better understand the environment. And so let me give you an example of this. Let's assume, all right, let's take a young child who's walking around with the caregiver. Caregiver might point to a poodle and say dog. Might point to a German Shepherd and say, that's a dog. And at that point, the child will develop a schema of what a dog might be. So it's this mental structure. And, it could be, for example, initially, that the child is going to come up with this idea, with this mental structure, that an animal that walks around on four legs is a dog, right? So it's a rudimental, at first at least, it has to be a rudimental way of understanding the world around them and organizing knowledge. So whenever, let's say, the child at this point walks around with the caregiver and they see a Labrador Retriever, right? That child might point to the lab and say dog, right? And the caregiver will say, yes, good job, that's another dog, that's a dog, right? So you see, that would be an example of assimilation, where the child is responding to new stimuli through their existing knowledge, their existing habits, their existing schema. However, especially initially, because their schema is so rudimentary, their mental structure is so vague, that accommodation is going to be required. So the example of accommodation would be that if that child now sees a cat, right? Cat being a four-legged animal, that young child is going to point to the cat and say dog. The caregiver would say to them, no, that's not a dog, that's a cat, right? So you see that at that point the child is going to have to change their existing schema, modify it, and create new ways of responding to objects, in this case to the animal. So they might further develop their schema through accommodation and say, well, a dog is a four-legged animal that barks, while four-legged animals that meows is a cat, right? So you see, accommodation is forming this new schema, modifying their existing schema in order to understand the world around them. according to Piaget, children go through a sequence of 4 stages of development. First, there is a sensory motor stage from birth to 18 months, 2 years. And description of this stage is knowing the world by sensing and acting, so by doing something physical and acting upon it. Like, for example, a young child is initially exploring the world mostly through their mouth, right? So they actually have to act on. So, for example, what I'm trying to say here is that if a young baby, get their hand on any object, what they'll do is that the first thing they do, they put it in their mouth, because their mouth is very sensitive physiologically and in terms of their sensory receptors, and will explore that object by feeling it with their mouth, right? So the idea that they only know the world by sensing, by sensation, and doing some action, doing something with or to their environment. The next stage is a pre-operational stage, approximately from 2 to 7 years, and that is the beginning of concept formation and symbolic reasoning, beginning of development of this higher order mental representation. We're able to categorize objects and some rudimentary basic symbolic reasoning. Next stage is concrete operational stage from 7 to 11 years, and that's where logical operation begins, but only on concrete objects and events. And finally, the formal operational period between 12 and going on to adulthood, and that's where abstract mental representation are formed, where a child is now able to use and understand analogies, metaphors, hypothetical reasoning. So you see the abstraction level here. Now let me point out some interesting aspects and supporting evidence of these stages, right? So beginning with the sensory motor stage, for example, remember how Piaget suggested that a child knows the world by sensing and acting, and that is supported by the idea of object permanence, that a young child does not have object permanence. Object permanence is the idea that whenever we see an object existing in the world and then it disappears from our view, we know that it still exists. But a young child doesn't have that ability. So when you take an object away from their sight, they act as if the object doesn't exist anymore. So out of sight is out of mind for a young child. So in this case, for example, in this slide to the left, the child exposed to a monkey over here, a toy monkey, will try to reach for it, then grab it. However, if you put a screen in front, the child will lose interest and behave as if they don't realize that that toy is still there. The next stage, the pre-operational stage, shows some interesting lack of ability. The fact that children at that stage, remember between 2 and 7 approximately, use centration, which means focusing on one particular variable at a time. So they cannot mentally process 2 or more aspects of a situation at the same time. And that is evident by looking at the conservation task. Because during a conservation task, a child is first exposed to two containers that are identical and asked to determine, by pouring juice in one container and taking it off in the other, to determine whether at which point these two containers will have the same amount of juice in this case. Right? At some point the child is going to say, yes, these two containers are the same. Now, if you take one of the containers and pour this content onto a different shaped glass here, and then ask again the child, which one's more, they will point to typically the tallest one. So what they're exhibiting here is a lack of conservation, the lack in this ability to conserve, in this case, liquids. But the same thing can be done with matter, for example, using Play-Doh, or with numbers, like using, let's say, a number of candies that are spread apart or bunched together. And you might have experienced this with even with food. If you have two young children eating, let's say, macaroni and cheese together, and if you give one of the child the food more spread out, and the other child the same amount of food but all bunched up on a single mound, what the second child might do is to complain and say that they want more, and all you really have to do is to take a spoon or a fork and spread that food out, and magically to that child that food would have become more. Now they're all of a sudden they're happy because they have the same amount as their friend, because it's more spread out. See, they're unable to do is to consider two variables at a time. The fact that you have to consider the relationship between the width and the height at the same time in order to determine the volume of whatever it is that you're considering.